Hi, we are going to be working through a Gizmo, which is a virtual lab, to explore different cell types. And I am going to go through this whole experience with you so that you can understand how to work through it. There have been a few issues, so just in case you can't get into the Gizmo, I'm going to go through it with you so that you can use this video to find the information. I haven't actually done this whole Gizmo before, so there might be some mistakes that I'm making so that it can kind of show you how sometimes you just have to play around until you figure something out when it comes to technology. So in the first part, I'm going to read that it says to go to the landscape tab, which is where I am, and it says click on the Elodia leaf. Where is an Elodia leaf? Something lit up. Maybe it's down here. There it is, Elodia leaf. What does it tell me to do with that? It says switch to the microscope tab. So here's the microscope tab. And observe the sample as it would appear under the microscope. So here it is. It looks awfully blurry. So am I supposed to do something about that? It says drag the coarse focus. Where is that? Here's where it says coarse focus. Drag the coarse focus slider until the sample is focused as well as possible. Then improve the focus with the fine focus. Okay, so I'm going to move this coarse focus. Is it getting, it's getting blurrier that direction. I'll go this direction and see if I can get it better. Ooh, it looks a lot more clear. Pretty good. Now I'm going to use this fine focus, see if I can make it any better. Ooh, not better. Not better. Perfect. Okay. That looks really good. That's cool. I can see all the little cells in there. I can even see some little green dots along the side, which is one of the questions on our sheet. It says, what do you see? So you could just describe that. And then it says to select the 400 times magnification. So there's 400 times. Now what do you see? Oh, cool. Look, I can see all these little green structures inside. And since I know it's a plant, and I know that plants have chloroplasts, and those are green, and the little things I see on here are green, I'm going to guess that I can actually see the chloroplast in these. So let's see what the next part says to do. Oh, the next part is about microscope parts and what they do. So, this says to go over, which I don't see that on this part, so maybe I'm going to need to go over this with you. So it says here's the stage up and down. Let's see. Oh, that's showing me the scales. This is showing me labels. Oh, this is labeling and showing me what everything is. That's cool. But this part on your worksheet is asking you to label the parts of the microscope. So maybe we need to go through those together. Here's the eye piece. Notice that that is where you would place your eye on the microscope to look through. Then we have B, which is the stage. That is the part that we lower or raise in order to better focus. Then we have, actually, I don't want to say that incorrectly. The stage is where we're going to be holding the slide. The fine focus is what moves that stage up and down, but the stage we aren't actually specifically moving. We're going to use the coarse focus. So this bigger knob is the coarse focus that's going to move that stage up and down so that we can see more clearly. And then we have fine focus, which is going to do the fine tuning of letting us get it as close and clearly as possible. And then what else? Oh, and then we have our objective lenses. So see how here it says 40, 100, and 400? Those objective lenses, we would turn these to go from one objective to the next. I wonder if it actually does it. Look, it does. Isn't that cool? There's the 100 and there's the 40. So the shortest one is the 40. This longest one that gets us up even closer is the 400. Okay, so now it says, with the 40 selected, use coarse and fine focus sliders to focus on the sample. Then choose 400 and do the same thing. Oh, and I'm sorry. Somewhere up here it said, I guess, to use skin cells. So let's go back and find a skin cell. Let's go back to our landscape. Here's a person. I'm guessing that's her skin. Oh, that's blood. Cool. That's muscle. Where's her skin? Skin. There we go. There's the human skin. Okay, so now I'm going to go, let's make sure that's right. Yep, human skin cell. Let's go to the microscope. It is not very clear. Let's start off on the 40. Oh, more blurry, more clear, more clear, more clear. Let me try my fine focus. Can I make it look any better? That's pretty good right there. So under 40, which focus knob is easier to use at 40 times is the question. Well, you kind of saw how it was for me to use it. Didn't seem too difficult. Turn on show labels. Oh, it won't let me show labels. Interesting. Oh, maybe I have to be at 400 to see labels? Oh, look. See? This is how it is when you do something new with technology. You kind of have to just work around with it. So here I am in the 400. Is it easier if I use my course? Oh, wow, that immediately gets blurry. Oh, it's hard to use that course adjustment at 400. The fine focus is definitely the better way to go. Okay, now I'm going to do my show labels. What do I see? There's the nucleus. I can see the cell membrane, and I can see all the cytoplasm, that light pink inside there. Then it says to turn off the show labels and go to the show scale bars. The show scale bar shows 20 micrometers, and let's see about how long it is using the scale bar. How do I manipulate that? Well, I would think that I could move that around a little bit, but maybe I'm just using this kind of as an estimate. So it's, if I were going to use this and go straight across, it would be about 20 micrometers. So I think that would make sense to put that down. Let's move on to the next page. It says, observe an organelle. Observe the samples below under the highest magnification. Click the show labels and label the organelles. List the organelles and approximate size of the cells in each sample. Okay, so here we go. Let's go back to this landscape. The first one is mouse skin, fly muscle. Where's the mouse? I don't see him. Oh, there's a little mouse. Okay. So there's our mouse skin. Let's get this more focus. It's always better to start off with the smallest focus before we go to the higher, because then all we have to do is use our fine focus, but it already looks pretty good. So let's do our show labels. We have a nucleus, a cell membrane, and cytoplasm. So that's what we would write. These would be our 3 organelles we would write in. Then our estimated size, it looks like one of them wouldn't quite be a full 20, right? It would almost be half, a little more than half. So maybe 15 micrometers, that could work. Okay, now we're going to go back and do a fly muscle. Where's the fly? There's a fly's muscle. Let's check that out. Microscope. Okay, so coarse focus adjustment, there it is. I'm going to go up to the 400. Let me turn off the scale bar so I can see. Okay, I have a cell membrane, I have a nucleus, and I have the cytoplasm. And then this is showing a striation. That's not really an organelle, it's just showing us kind of the differences in the cells in there. And then I'm going to do my scale bar. And this muscle cell is really large. So it's, if we go from this section to this section, and if we think about how far this is, I wish we could turn it. I wonder if I'm just not doing something correctly, but that's okay. So if we turned it, it would be pretty large. It might be 40. It might be 2 of those going across. So I would say that one's about 40 micrometers. Now which one do we get to go to? Go back to the landscape. We have a maple leaf. Here's our maple leaf. Let's go to the microscope. Coarse adjustment. There we go. That looks cool. 400. Let me turn off the scale bar. Okay, so knowing this is a plant, I'm not surprised to see a cell wall as an organelle. cell membrane. There's our nucleus. We have our cytoplasm, and we can see this nice large vacuole. That all makes sense for a plant. And then if we look at our scale bars, that one doesn't look like it's a full, this one looks larger than this one. It's kind of a variety of sizes, but it doesn't look like it's the full 20 micrometers. It's probably more like 12 micrometers, I would say. Okay, back to that Elodea. That's what we started with. This is a water plant, so it might be a little different. Nope, I'm going to have to go this way. Fine focus. Go up to the 400, turn off the scale. Okay, so in here I can see the chloroplasts. That's what's cool about those water plants. You can actually see the chloroplasts. It's hard to see them in the maple leaf. So chloroplast is an organelle. We've got our nucleus. We can barely see it right there. Our large vacuole, the cell membrane, and the cell wall. So that's a lot of the organelles we're able to see in that one. Then if we look at our scale bar, oh, that's a fairly large cell. Probably 2 of those would fit. So I'd say about 40 micrometers. And last one is the fungus. There's a fungus among us. Okay, let's see what we have to do to see it. Whoa, whoa, I saw it. There it is. Let's make that one bigger. Might need to focus a little bit better. Fine focus. Let's get better. There we go. Turn off the scale bar. So we have a cell wall. So like plants, a fungus has a cell wall. We have our cell membrane. Ooh, there's a fairly decent sized vacuole and a nucleus. Oh, and there's also a cytoplasm. They just use this part to show that. So there's a cytoplasm. So let's turn on our scale bar. So if just this one section, this is one cell of a fungus, what size do you think that would be? Maybe about the 10 micrometer? I think that would make sense, a little bit smaller. Okay, then we have some questions to answer about that. And then it goes on to click on the cow and observe the E. coli. Okay, click on the cow. E. coli. What are we going to check out about the E. coli? In the E. coli, let's do our focus. Oh, they're little. So E. coli, what in the heck are these things? Oh, look, we can go all the way to 1,000. So we're really getting magnified in on these guys. So there that is at 1,000 and then 2,500 magnification. Oh my gosh. So this has a cell wall, cytoplasm, and a cell membrane. But guess what I don't see? I don't see a nucleus. That makes me think this is probably a bacteria. Also, because it's so incredibly tiny, that also makes me think that it is a bacteria. The questions about it, we already went over the organelles. Oh, we already went over nucleus is missing. And then the approximate size, this is 4 micrometers. Oh my goodness, and it's not even, maybe half of it, only 2 micrometers. So it's much, much smaller. Okay, so at the end, look at the sand silt sample under the microscope. Where is that? Sand and silt. That's a frog. Protists, those would be cool to look at. Microalgae. Sand silt. There we go. Okay, does it have internal structures? Can we see anything inside of it? No, nor organelles. Would that be a living creature? Even up close? No, we don't see any organelles inside of it, so it is not living. And that is the end of all you get to do on this. So I hope you went through this with me and you enjoyed it. Feel free to go back and review any part of it because you should be able to answer all the questions just by watching this.